So our first speaker, I want to brag about her a little bit. Our first speaker is Dr. Karen Zito. She's a professor of neurobiology, physiology, and behavior, and a core faculty member at the Center for Neuroscience. She got her Ph.D. at UC Berkeley and did her postdoctoral training at Cold Spring Harbor Laboratory. She's won numerous distinguished awards, including a Burroughs Wellcome Fund Career Award, an NSF Career Award. She's been selected as a UC Davis Chancellor's Fellow, and in 2021, she was elected as a Fellow of the American Association for the Advancement of Science. In addition to her dedication to research, Professor Zito is passionate about teaching and mentoring, both in the classroom and in the laboratory. She currently serves as the director of the NIH-funded T32 training program in learning, memory, and plasticity, and she has served as faculty and directed courses on microscopy, both nationally and internationally. Professor Zito, as you'll hear directly from her, she studies synapses, the connections between neurons where plasticity occurs. Her research is focused on dendritic spines, the tiny protrusions that extend from the postsynaptic neuron and are highly dynamic, changing size and shape as rapidly as the synapse grows and shrinks during plasticity. An important goal of her current work is to identify the molecular signaling pathways that are active when the synapses undergo these changes, and by identifying these pathways, she hopes to better understand the cellular and molecular mechanisms of how we learn, which may help to identify targets for therapeutics when learning is disrupted. Karen.
Thanks, Marie, for that kind introduction. Can you all hear me? All right, let me pull it up a little bit. How about that? Better? All right, yeah, need my collar today, but. All right, so I'm really excited to be here today to talk about the research that's ongoing in my lab. As Marie mentioned, my lab is really interested in what changes in the brain when we learn new things. And in particular, what we're very interested in is identifying, we're cell and molecular biologists, so we want to understand the molecular signaling mechanisms that are important for learning, what molecules are changing, how are they involved, and determine how they're altered with diseases that disrupt learning, with the hope eventually that we could develop therapeutics that could help people learn that have trouble learning. So we study this in a part of the brain that is, actually maybe I should use the pointer because we study this in a part of the brain that this blue part up here, the folded part, the cerebral cortex, it's a part of the brain that's really involved in the higher order processes like perception, cognition, learning, and memory. And we're studying neurons and how they change when we learn. And this is just a little plug of the cerebral cortex. You can see there's tons and tons and tons of neurons there. Our focus is on one particular really important type of neuron. It's called the excitatory pyramidal neuron. And you can see here, it's got a lot of really extensive processes. These are called dendrites. And they're the sites where incoming information, they collect all the information from other neurons. And it's these dendrites, if we zoom in on them, they're studded with little teeny protrusions called dendritic spines. These are microscopic little protrusions from the dendrites, and an axon will pass by, and it'll make a synapse onto the spine. So the axon has vesicles of chemical neurotransmitter that get released onto the receptors in the spine head. Those receptors then open, let ions through, and that create an electrical signal that powers the circuits that drive our behaviors. And so what we're interested in, the cellular theory of learning, is there's a way that those synaptic connections are changing that allow us to learn new things, and we're interested in understanding that. So there's two main ways to look at how these connections may change. One is, I showed you an existing connection. It can get stronger or weaker or grow and shrink. Another thing that can happen is the pattern of connections changes. Neurons that weren't connected before can become connected, and neurons that are connected can become disconnected, and that can help us learn. We're interested in both of those processes. So we want to peer into the brain and see what's happening. We couldn't do this a long time ago, but more recently, as Marie mentioned, there's a lot of technologies that have been developed over the past couple of decades that have allowed us to peer inside the brain and check out how learning is happening. One thing that happened is that we're now able to genetically label neurons, and I'll tell you about that in just one sec, but that allows, our neurons aren't glowing, they're hard to see. We can't just look inside the brain and see our neurons, we have to label them to see them and see what they're doing. Another thing is there had to be a microscope that would allow us to peer deep in the brain, and a 2-photon microscope is that type of microscope, and that was invented in the late 1990s, so it's only been about 25 years that we've been able to do this peering into the brain. So as for genetically labeling, many of you may have heard of this green fluorescent protein before. GFP, how many have heard of GFP? Significant number. Yeah, so GFP was discovered way long ago in the 1960s by Art Shimomura, and he was studying jellyfish. So basic neuroscience research, he was studying, well, what is it that allows jellyfish to glow? You might have seen those jellyfish in the Monterey Bay Aquarium if you've seen them or in pictures. They glow, and he wanted to find out how do they do that? What protein is responsible? And so he cloned the gene for GFP. And it took a little while, but people started to realize, Marty Chaffee, who worked in Columbia University, realized, well, can't we take this gene and insert it into mammals and cause them to glow, their neurons to also glow? And so he did. He transferred that GFP molecule into neuronal cells. He did it first into C. elegans. This is a worm that now glows, its neurons are glowing, and then later into mice. And it's interesting, many labs have been working to optimize these fluorescent proteins and allow us to have a toolbox to use to do different things. One person was Roger Chen, who was at UC San Diego. He worked on modifying these proteins so that we have many colors now. These are all the different colors that he created. This is way long ago. We have so many different tools now. So all of these three guys won the Nobel Prize for their work on green fluorescent protein. So we use that protein, and it can be inserted into the mouse brain and label a small subset of neurons that allow us to image inside the living mouse and see what's happening when learning happens. And so this is some early work from Josh Trachtenberg, where the mouse, what he did was he took a little piece of skull off and put a cover slip on so it has a glass window into its brain. And it survives for the whole lifetime, just with a little glass window. And then he can look at the same neuron over and over when we're learning and see, well, what is happening? And here's a GFP labeled dendrite from this neuron, and he sees that, yeah, from day 1 to day 2, there's these blue spines that are staying the same, but these red ones are changing. So when he quantified how many days a spine lived, he had 8 days here, he saw that most all spines fell into 2 groups. Either they were living all 8 days, like these blue ones, you can see that they're living the whole time, and those, he said, those are probably stable circuits. Those are connected to axons and involved in circuits that the brain we're using to behave. And then you can see these red ones, they come and they go, they come and they go. And these, he thought, were like substrates for plasticity, searching for synaptic partners and saying, well, is there an axon that will help me learn something new, like help support learning? And these were transient or learning spines. And the idea is that with learning, one of those transient spines hits an axon, makes a new connection that makes a new circuit, and then the circuit becomes established that allows us to do a new behavior. And so we went looking for these. So this is other labs now too. Another lab at UC Santa Cruz this time, Yuzu's lab, was looking at rodents to see, well, she's peering into the brain imaging while the animal's learning to pick up pasta. No kidding. Capellini are actually hard for a rodent to pick up. And she's trying to see, it's learning how to pick up this pasta. And she sees new spines grow out when it learns something new. So there's a new spine, and you can see here, when it's learning to pick up the capellini or the pasta, it grows new spines. And that's a change in connectivity. It's growing a new spine to make a new circuit, we think. Another thing that happens is, I don't know if you know, but when you lose a particular sense or a limb or something, the remaining stuff will take over the brain, will start taking over the brain. So if you actually put a cover on one of the eyes, the other eye will start taking over the brain. And so when she did that, she saw that new spines would grow, but then if the cover was removed, they would shrink, they'd get smaller, and then if the cover was put back on again, they'd grow. So not only do they grow out, but they shrink and grow based upon whether they're being used. And so that's a change in that represents a change in synaptic strength. So the last thing I want to tell you as background about spines is that there's many diseases that spine densities are changing with aging and with disease, and times when there's altered intellectual performance. And so this is just a wild type animal, a normal human. So essentially, early in development, we have a ton of spines, synapses made, and then as our nervous system matures and gets formulated and matched in the environment, there's pruning. And then during adulthood, there's 2 lines here. So that's our whole adulthood. We have a pretty even spine density. We have both growth and retraction. And then when you start reaching a little bit later, around my age, probably those spines start going away a little bit. Yeah. But during disease, like in schizophrenia, there's a dramatic decrease in spine densities that are happening during adolescence with the onset of symptoms. And also in Alzheimer's disease, there's a dramatic decrease in spine densities that associated with the disease. And so, again, my lab is really interested in this question: can we identify the molecules that are controlling the stability and the outgrowth and the stabilization of these spines so that we can potentially identify targets for therapeutics to help block their loss associated with disease and help people learn, we hope. Okay, so what kind of questions does that lead to? Well, we have a dendrite, but we need to grow a new spine, so what molecules initiate the growth when we're making a new circuit during learning? How are spines stabilized? Once we grow a new circuit, we know we need to connect to an axon and make a new circuit, so what molecules are involved in that? And then how is there a balance? We know in our brain we're constantly learning new things as adults even, but our densities don't change, so there's some kind of balance, and so we need to know which spines to keep and which to get rid of. So we're interested in the molecular mechanisms that drive all of those questions. So for our methods, my lab is working mainly in vitro, in neurons that are in vitro in the dish, and we're labeling them with GFP, so we can actually use 2-photon microscopy, again, to time-lapse image and watch new spines to grow and watch them stabilize. We can also manipulate them. So there's a technique called 2-photon glutamate uncaging where we can bathe the slice in a glutamate that's been prohibited from being active chemically. Then light can release that chemical, and we can activate individual spines and individual synapses by releasing excitatory transmitter. And so here you can see there's a red calcium-insensitive dye and a green calcium-sensitive dye, and if we uncage glutamate right next to a single spine, calcium enters into that spine, so we can manipulate one synapse and not the other nearby. We can also put a patch pipette, a glass pipette onto the soma to measure electrical activity, and we can measure how the neurons send electrical signals in response to inputs at single synapses. And if we take those techniques and we can manipulate genetically molecules, either upregulating or downregulating them or blocking them pharmacologically, we can identify the molecular pathways that are involved in these processes, these spine growth and stabilization. So I'm going to tell you just a really brief summary of one project that we're doing that's related to the theme of the symposium today, which is how can synapses be both plastic in order to encode new memories and stable in order to retain old ones. So in our lab, again, there's a lot of history behind long-lasting memories in our brain we think are associated with really large, stable spines. And as I mentioned, those ones that turn over rapidly are not stable. But what's really interesting to us are these memories we're just starting to learn. Let's say we're trying to learn to ride the bicycle, and we're learning a new memory. And then at the same time, once we're done with that, we go in and we learn to play the piano. Well, we don't want the piano circuit to override the bicycle circuit, right? So how do we do that? One hypothesis is that those bicycle synapses that just got strengthened when you were practicing it, maybe they're protected from further plasticity while you're doing your piano practicing for a brief period of time. So we were interested in testing that out. And so this is an experiment that Juan Flores, a project that he led in the lab, and he's here I would highly encourage you. He's heading the microscopy section, the booth over there. If you want to talk to him about this research, he's fantastic. Just recently published this paper last year. So he proposed that, well, maybe a synapse that recently was incorporated into a new circuit that recently had plasticity, maybe it was refractory to new plasticity. And so how did he test this? His approach was, well, he would stimulate using that glutamate uncaging I tell you about to make a synapse bigger and long-lasting, more stable and strong. And then he checked again just a short period later to see if that same synapse was able to still undergo plasticity. And he didn't know, well, would it be refractory or would it also grow? And on the same cell, he stimulated a different synapse that hadn't had a prior stimulus and looked to see if that one grew, and it should. So this is just his data. So again, here he does this local uncaging on one spine. The neighbors are all flat, they don't do anything, and he stimulates it, and he sees, boom, this transient increase and then a long-term growth. So the synapse is stronger and more stable. He tries again 30 minutes later. It responds just a little bit, but it doesn't grow at all long-term. So this synapse, that for the exact same stimulus, that it was able to undergo plasticity is no longer able to undergo plasticity. And he did a control spine on the same cell, and that was easily able to do a long-term strengthening that would be equivalent to a learning type event. And so it's not the whole cell has changed, just one synapse that recently underwent plasticity was unable to do it again. So Juan wondered, well, how long does it take for it to recover? Like, how long does it hold on to its refractory bit? And so he said, let's try 60 minutes. So here now he stimulated, saw the long-term growth, stimulated again, and saw long-term growth. So these synapses are protected for about 60 minutes from undergoing an additional types of plasticity. And again, it was perfectly recovered. It looked just like an unstimulated synapse. So Juan was wondering, well, what molecules are changed? Why does, when we stimulate the spine the first time, it grows and makes a stronger synapse, and then the second time, just shortly after, what changed? What is different? And so he went to look at molecules that change after plasticity. So there's a lot of molecules that are known already to be active at the synapse. And we know that one of those plasticity molecules must not be changing fast enough after stimulation to recover the plasticity. And so he looked at this molecule Ph.D. So here, all these ones up here, you can see there's the green tagged protein, it changes, and the red tagged spine volume, and they all change in sync. But for down here, you'll see there's a bunch that also don't change in sync. And so we thought these would be good candidates for ones that might need to be around in order to induce plasticity again. So Juan went, and what did he do? He went to look to see, well, what if I increase the level of one of those proteins that's not there fast after plasticity happens? It kind of goes away from the synapse. And so he added a little bit of extra Ph.D. back. You can see that in green. And he stimulated the synapse. He got the long-term growth, and then he stimulated again at 30 minutes, and he had recovered long-term growth. So that molecule was enough to help the synapse recover. And this is just showing that it's fully recovered, just like neighbors. So Juan was like, well, I gotta show people that it's not just any molecule. Is it every single synaptic molecule that would recover this? So what he did is he took a molecule, Ph.D. which is only a little bit different than Ph.D. He overexpressed that, and he got, boom, plasticity, long-term growth, synaptic strengthening associated with learning, but the second stimulus showed no change. So he was able to identify one molecule that looks like it's absolutely needed for our synapses to undergo plasticity that's important for the refractory period after learning. So what Juan was wondering is, well, why can two molecules, these are the two molecules drawn out in schematic. They look really similar. In fact, in this schematic, you can't even tell the difference. They're so similar, Ph.D. and Ph.D. So what is it? Why can Ph.D. restore but not Ph.D. And so what he saw is in the literature, they interact with, even though they're so similar, those very few changes in amino acids cause them to interact with different proteins. And so we have to identify what proteins are involved in this whole pathway that allows us to regulate whether a synapse can be plastic or not. So the interesting thing is also in the literature, it's been shown that the two, even though they only have a few amino acid changes, can behave really differently. And some other exciting new steps that we will be doing as well. We've started a collaboration with another lab where we're gonna watch these synapses grow when they're making a stronger, bigger synapse, and then when they're refractory to new growth. And we're gonna look at the ultrastructural level. We're gonna zoom in using electron microscopy to check to see what's happening at the synapses exactly when they're refractory to plasticity. So what did I tell you during my talk about how brains change when we learn new things? I told you that connections between neurons in our brain occur on these tiny protrusions called dendritic spines, shown here. I showed you that growth of these new dendritic spines is associated with learning new things. And so we think that's the basis of the circuits that are forming that underlie learning. I discussed that these spine synapses are often altered with aging and in disease, so we lose them with schizophrenia, we'll lose them with Alzheimer's disease, and so we really want to identify this molecular signaling pathways that lead to the stabilization and the formation of new synapses when we learn, so that we can target them for therapeutics to help people learn that have challenged learning. And then finally, I talked about the theme of today, which is, it looks like a refractory period for spine synapses exists that may provide an important mechanism to prevent overwriting of recently learned things, like when you're riding your bicycle and playing your piano at the same time. And it also could contribute to the advantages of spaced learning. So you may have heard of this, but it's actually better if you do little bouts of learning and take a rest in between. So you learn, practice a little, take a rest, practice a little, take a rest. We think in those rest periods, it's the time period that those molecules are recovering that need to be around to help us learn new things. So finally, I'd like to thank my lab, and in particular, this project was led by Juan Flores, and like I said, he's out there at the microscopy display if you'd want to meet him. This is my lab, and then I'd like to thank my collaborators, Ryohei Yasuda and Kristen Harris on this project, and then funding from the NIH, and I'd like to thank you for your attention, and I'm happy to take any questions.
Thank you. Yes, I wanted to know the process of learning and remembering. Does it work the same way when you have the process of learning to forget something that doesn't apply anymore? Does it work the same way, or is it does? So that's just learning something new again.
No, that's a great question. I was laughing because it's such a great question, and I don't think we have much knowledge yet about how, when we're trying to forget things, exactly what's happening yet. But there are definitely some experiments out there. So for instance, some of these mouse experiments where the animal's learning, say, to pick up a capellini. It turns out that a lab in Colombia did show that a similar type task that they were learning, when they actually trained the animal to forget, those same dendritic spines that had grown did go away for that task. But it's interesting because in some other cases, that's why I was laughing, because it's such a great question. In other cases, it looks like getting rid of that old memory involves forming new connections too. And so, what's that? There's no just delete button, apparently. But 1 or 2 labs have shown that does happen sometimes, which would be great. Yeah, an easy explanation. Yeah.
Why did you choose to go forward with Ph.D. 95 over any of the other molecules that showed a difference between?
Yeah, so Juan chose Ph.D. 95 because I didn't get to show you all the data, it's a short talk, but its time course matched most closely the time course of recovery. And so that's how he chose Ph.D. 95 above the other ones. The other thing is Ph.D. 95, I'd like to tell you 3 more things about it. It is known It's a molecule that's structurally important for the synapse, so it seems to link together a whole bunch of molecules that are important for stabilizing spines. And that's another reason it seemed like a good candidate. And a third thing is that it does look like there's reports that with aging and Alzheimer's disease, there's a decrease in amount of Ph.D.95 that's associated with those diseases. And so that suggests it could be a good candidate for checking out, yeah.
Does that newly forming spine make connections just with axons, or does it make connections with other spines as well?
That is really interesting. You know, in science, one learns to never say never. It's very rare, but it definitely, there's some instances, especially in the olfactory system, where we have what's called dendritic synapses, which is where a spine connects with a spine. And so that is common in one particular part of the brain, but in our cortex, in our cerebral cortex, I would say that's almost never observed. It's very rare. It's really a one-way communication, an axon going to a spine in that particular area. Yeah, really interesting. Yes.
Hi. I'm fascinated with your work. You're working at a different level than I am, and I'm just really curious about if you can make an enormous jump to children who are who typically cannot learn in the same way as other children, like children with learning disability, children who have attentional problems. And if there's any work being done at the molecular level or this level that can generalize to that? How's that for a big question?
Yeah, no, I would say definitely yes. I feel like when we're developing therapeutic targets, a lot of what'll happen is the first stage will be, oh, can we recover, let's say we have altered synaptic plasticity in some model for disease. And the first thing we'll do is, oh, can we recover how these synapses are growing and stabilizing? But then the next step would be, oh, can we recover how the problems with the animal's behavior? So that would be the next step. And then we would think about moving into humans if that worked well. And so it's really interesting. Again, this is something that's been coming up more recently. Mice are a fantastic model when you're looking at a really small region, and we learn so much. But they're not everything, right? And so there's molecules that will succeed in mice, but then fail in humans. And so we're trying to develop new approaches, whether it be computational or whether it be using, more advanced, different models, or whether it be actually using human neurons. I don't know if Martina's here, but to see, oh, there she is, to see if they respond the same way, just to give us a better sense before we would go to those clinical trials. Yeah.
Last question.
Hi.
Mechanisms behind normal learning in a healthy brain, of neuroplasticity are the same as neuroplasticity after cell death from a stroke or something?
I didn't hear the very first thing, but stroke I heard. Can I address that one? Yeah. So the question is the plasticity mechanisms, are they same that are happening just when we learn new thing as what happens after stroke? And I would say no, or traumatic brain injury. I would say that after those type of really extreme changes, that essentially sends you down a completely different pathway that involves some of the same molecules, but usually stroke and TBI, traumatic brain injury, involve a much larger change in large-scale neuronal connections. So what you saw from me today is I'm talking about those teeny little synapses. Those spines are growing in and out, but we're talking microns. Your brain is normally changing only a little bit. It makes a huge impact if different neurons connect with different neurons to make different circuits, but it only involves a small change in actual anatomy. But when we talk about traumatic brain injury or stroke, we get new axons growing and new dendrites growing because there's a lot of cell death. And that would be a very different process there.
Thank you so much, Dr. Zito.